Good evening, everybody. And I would like to welcome our honorable speaker, Dr. Sanghamitra Bhattacharyya, who is joining in this webinar, and she is ready to deliver I am sure that it will be very much enriching and entertaining lecture, and our students and our colleagues will be able to know so many things which are really new to us, and you are joining from a different part of globe, so we are really glad to meet you, and I'm sure that this webinar will be a successful one. And now I would like to request Departmental Head, Department of Botany, to carry on the webinar. Thank you.
Thank you, ma'am. On behalf of the Department of Botany of Narasingha Datta College, lot of thanks to our principal ma'am, Dr. Shoma Bandopadhyay, for the inaugural address of today's international webinar. It is my great pleasure to welcome all of you and also today's speaker, Dr. Sanghamitra Bhattacharyya, in this webinar, which is being jointly organized by the Internal Quality Assurance Cell or IQAC of the college and Department of Botany. Dr. Sanghamitra Bhattacharyya, Senior Research Scientist of Lerner Research Institute at Cleveland Clinic, Ohio, United States of America, did her graduation and post-graduation in botany from Vishwabharati Shantiniketan. She worked as a research fellow at Bose Institute, Kolkata, and received her PhD degree in botany in 1999 from University of Calcutta. Her doctoral research work was based on plant tissue culture and biotechnology of medicinal plant Nyctanthus arbortristis. She was engaged with postdoctoral research on plant molecular biology at Bose Institute till 2006. Further, he continued her research on molecular biology in Cleveland Clinic Foundation, USA. She worked as postdoctoral research scientist in Case Western Reserve University, USA, for 8 years. Presently, she is attached with Lerner Research Institute at Cleveland Clinic, Ohio, USA, and during her research career, she had published a large number of research papers in the journals of national and international repute. These are just few words to tell about today's speaker, today's research person. We all are eagerly waiting to hear from her. Thank you, ma'am. Welcome, Dr. Sanghamitra Bhattacharyya. Over to Dr. Sanghamitra Bhattacharyya. Madam, please unmute. We can't hear you.
Give me a second. There is a glitch here.
Yes, you are audible, ma'am.
Yeah, but the sharing, it's taking some time.
Yes, your screen is also visible.
Screen is visible, but why is it here? It says stop presenting.
It's not a problem, ma'am. Please carry on.
Okay, give me 1 second. There is some problem from my side. 1 second. It's stuck here. Yeah. Is everything okay over there at your side?
It's all right, ma'am.
Okay. Namaskar. Good evening to all my attendees, and a big thank you to a very interesting introduction. I thank the faculty members, the students, friends from near and far who are attending my talk, my respected teachers, and of course, my lovely family members. I earnestly thank the staff of Narasingha Datta College and particularly the Department of Botany to give me this opportunity to share a part of my research. Why do we age? Why does cancer develop? What's the connection between heart failure and Alzheimer's, or infertility and hearing or vision loss? Can we extend our lifespan? And if so, how can we do it? And many, many more questions continue piling up. What I think as a basic science researcher is that all these questions can be answered by understanding this very interesting organelle, mitochondria. And from this point of view evolves today's topic, mitochondria inspire a lifestyle. Before I proceed any further, I would like to take the opportunity to acknowledge my lab for their wonderful contribution, and particularly Dr. Vera Bonilla, my advisor, my colleagues Mala, Jonathan, Elise, and Paul for their cooperation all the time. Now, one time, while I was trying to analyze my research findings, I came across a book named Mitochondria and the Future of Medicines, written by Lee No, a very famous naturopathic doctor from United States and a big time of Star Wars fan. Now, you all might be thinking, what does Star Wars have to do in this talk? And what's the connection to mitochondria? Now, those who have watched the movie know very well that in Star Wars, there were characters called midichlorians, which were microscopic life forms and symbiotic in function when inside the living form. These midichlorians were energy providers to the functional Jedi characters. According to the author, there lies the connection. Like the midichlorians who provided energy for the fictional Jedis, mitochondria are energy providers of this living planet. These midichlorians are man-made, but who made the mitochondria? Let's go back billions of years ago when one of our single ancestors engulfed a unicellular prokaryote. Now, these prokaryote and the actual cell, both these two cells now knew that they have to grow and reproduce together. They have to adjust, no fighting. The prokaryote performed aerobic respiration and generated ATP. The endocytosed organism ultimately evolved into present-day mitochondrion. Here lies the other similarity according to Lee No, the writer, with the midichlorians. Both of them, the midichlorians and the mitochondria, they started life as a single form and then joined another living form and finally became a part of togetherness. So mitochondria, according to him, can be referred to as the midichlorians of this non-fictional living world. The modern day mitochondria still have their own circular DNA, and every mitochondrion contains multiple copies of its own genome. The powerhouse of the cell, or the battery pack, is a very interesting organelle. In humans, mitochondria contain around 1500 proteins, of which the 13 proteins are encoded in the mitochondrial DNA. Most mitochondrial proteins are synthesized via cytoplasmic ribosomes. Proteins that are key components of the electron transport chain are exclusively translated in the mitochondria. Now let's see the different parts of this interesting structure. First, to start with, are the mitochondrial ribosomes. It's an active protein complex in the mitochondria having two subunits, a large and a small subunit. You see the small red dots here, they are the mitochondrial ribosomes. scattered on the matrix of the mitochondria. Now, the other important function of these ribosomes are it translates mitochondrial mRNA encoded in the mitochondrial DNA and finally helps in ATP production. The matrix of the mitochondria, this light-colored structure, is the internal space enclosed by the inner membrane. So this is the outer membrane of the mitochondria, and this light, you see this boundary here of this light structure, that's the inner membrane. And between and inside the inner membrane and close by the inner membrane is the matrix area. It contains different enzymes and in high concentration, and hence it's involved in the different steps of cellular respiration. Then comes the cristae, which are these invaginations of the inner membrane, and it increases the surface area of absorption. And because of this, it's also involved in different important chemical reactions during ATP synthesis, and it also helps in aerobic cellular respiration. I'll explain this part a little bit more in the next couple of slides. The other important structure that we see in this organelle are porins, which are beta barrel proteins cross a cellular membrane. So this is actually, it starts in the outer membrane and it forms a tubular structure, a connection between the outer and the inner mitochondria, where it acts as a pore that helps important molecules to diffuse during oxidative phosphorylation. Like the mitochondrial proteins, chloroplast, also the outer membrane of the chloroplast also contains porins for the same reason. The major functions of the mitochondria, which is referred to as the energy currency of the cell because of its contribution, huge contribution, I would say, in ATP production. The other functions are calcium homeostasis, regulation of innate immunity, programmed cell death, and stem cell regulation. Calcium homeostasis and programmed cell death are also important functions that maintains mitochondrial biogenesis. Apart from this, the other most important function of mitochondria is it acts as a signaling platform that communicates changes between the mitochondria and the nucleus through retrograde signaling, or between the nucleus and the mitochondria through anterograde signaling. Let's check the story inside the organelle. Now, the glucose we derive by the oxidation of carbohydrates runs the process of glycolysis and forms pyruvate in the cytosol. Now, once pyruvate is formed, it enters the mitochondria and it is converted to acetyl coenzyme A inside the mitochondrial matrix. Acetyl coenzyme A then enters the citric acid cycle, also called the Krebs cycle, that produces ATP, reduced NAD, and many amino acid precursors. Now, this brown structure is the inner mitochondrial membrane, and located on this inner mitochondrial membrane are four complexes, 1, 2, 3, 4, you see it over here, and these are important components of the electron transport chain. These four complexes together, they create a proton gradient across the inner mitochondrial membrane, enabling the ATP synthase, the most important component in ATP production, through oxidative phosphorylation. Now, this ATP synthase is a multi-subunit membrane-bound enzyme of the mitochondria, which utilizes this proton electrochemical gradient generated by the respiratory chain for helping in the synthesis of ATP. Now, the activity of ATP synthase, there are some more in its functional points, and it regulates the flux of oxidative phosphorylation. It helps in the execution of cell death, also called apoptosis, and it also helps in mitochondrial signaling by ROS. ROS, or reactive oxygen species, can be created naturally through or as byproducts in the electron transport chain, and it has a tendency of damaging the mitochondrial DNA. Like ROS is produced naturally, nature has brought out Some help to protect the mitochondrial DNA. And these are naturally occurring antioxidants that help neutralizing the effect of this naturally produced ROS. These natural antioxidants are like N-acetylcysteine and coenzyme Q10. We have just mentioned ROS generation inside the active mitochondria. Reactive oxygen species, like I mentioned, is a natural oxidant, and the effect is neutralized by the natural antioxidants. But what happens if either the natural antioxidants are not sufficient, or they are not produced, have a problem in its expression, and as a result of that, ROS increases, which is not at all a good situation. If not removed, it accumulates and it gives rise to a stressed condition, which we refer to as oxidative stress. Oxidative comes from the term that these ROS are natural oxidants. So the stress produced by the oxidants are oxidative stress. So in one sentence, we can define oxidative stress as an imbalance produced by ROS production and elimination due to non-functional biological defense molecules, one of which are the antioxidants. When we talk about ROS, we hear about another term called free radicals. Now, free radicals are oxygen-containing molecules with an uneven number of electrons. And because they contain this uneven number of electrons, they always remain hungry to easily react with other molecules. Free radicals are formed from UV light, inflammation, normal metabolism, smoking, ionizing radiation, including radiotherapy, and air pollution, and has a tendency for damaging the DNA by breaking the DNA strands. Now, if we look at the left side of this cartoon, we see a healthy mitochondria happily producing ATP, providing energy to the cells. This is a damaged mitochondria formed from toxins, unhealthy food, stress, and it gives rise to a dysfunctional mitochondria and releases the free radicals. And finally, this toxic effect affects our body and results in health complications. Oxidative stress, interestingly, is not only for the animal world. The plant system also encounters ROS and gives rise to oxidative stress. Different types of stress are drought, air pollutants, soil salinity, heavy metals, biotic stress, high light intensity, and high temperature, resulting in male sterility, a low level tolerance of stress and cell death, variegation, albinism, and this all results in an overall retarded growth and development of the plant. Now, we have already been introduced to mitochondrial DNA a couple of slides back, and we all now know that it helps in ATP generation. But there are some other very interesting points about mitochondrial DNA. It's solely inherited from our mothers, and hence it's designated as the mitochondrial Eve. It's a very small circular chromosome, approximately 16 to 17 kb. It encodes 2 ribosomal RNAs, 22 transfer RNAs, and 13 proteins that serve as subunits of the respiratory chain. Interestingly, during evolution, many of these mitochondrial genes have walked into the cell nucleus. This has been shown for the first time by the Lerner Davis School of Gerontology that the mitochondrial and nuclear genomes, both, they co-evolved to independently cross-regulate each other. They are encoded in the nuclear DNA and are referred to as nuclear encoded mitochondrial genes. Now, these nuclear encoded mitochondrial genes, of course, gets translated into proteins that gets expressed in the mitochondria, and they have been identified and characterized by mass spectrometric based techniques. Now, these nuclear encoded mitochondrial genes tends to be older than the other duplicated genes in the genome. Mitochondria is inherited maternally, and a mitochondrial DNA test traces a person's mother line ancestry using the DNA in his or her mitochondria. But what happens to the paternal mitochondria? In humans, most of the paternal mitochondria reside at the base of the sperm tail, and upon fertilization, when the sperm loses its tail, it also loses most of the paternal DNA, mitochondrial DNA. If, however, the paternal mitochondria enters the egg during fertilizations, if they are able to, they are eliminated with the help of specific mitochondrial destroying enzymes. Now, even if some of these paternal mitochondrial DNA can overcome this second stage, the ones that manage to evade these enzymes would end up being extremely diluted, and because inside the egg we have around 200,000 mitochondrial DNA molecules, while the sperm contains an average of, it could be 5 or just 10. We have heard about the changes, that changes are inevitable and sometimes they are required for survival. Sometimes they are advantageous for our lives and sometimes they are not. To function correctly, each cell depends on thousands of proteins to do their jobs in the right places and at the right times. Each particular protein carries the messages from their genomic counterpart. So even a slight variation in the gene sequence will give us a totally different protein. These variation in the gene sequence are termed as gene variants, and we can also term them as mutation, and they prevent one or more proteins from functioning properly inside our body. A condition caused by these variants can collectively be termed as genetic disorders. So genes themselves do not cause disease. It's the genetic disorders that are caused by variants which can alter or eliminate a gene's function. And it's ultimately the proteins that arise from these genetic disorders actually cause the problem. Mitochondrial DNA, the mutation in this DNA, produce a malfunctional protein, or sometimes the protein is not produced at all. It disrupts normal development, compromising health condition, especially in multiple organ systems that require a lot of energy like heart, brain, and muscles. It can also prevent an embryo from surviving to birth. Mitochondrial DNA has a limited ability to repair errors and hence it builds up health defects. Non-inheritable somatic variants are also present in mitochondrial DNA. And since they are non-inheritable, it piles up, and this piling up of somatic variants does not seem good for the cell because it can cause cancer, increased risk of certain age-related disorders, especially Alzheimer's and Parkinson's. Mitochondrial diseases are caused by abnormal genes leading to flawed proteins or other molecules in the mitochondria. The various subtypes, here you see different diseases from different living organs inside our body. And the various subtypes are caused by alterations in different genes, leading to worn down cells in different parts of our body. So let's see, like if a gene from the nuclear DNA, if it has some sort of variation in it, it makes or it gets translated into a different protein. We don't want that protein, but however, that protein is targeted in the mitochondria where it was supposed, the actual protein was supposed to take part in oxidative phosphorylation to produce ATP, and then the ATP is distributed to the organs of the body for their sustenance. But once there is a variation in the gene, the protein that the gene gets translated into is not the right one, and hence this entire process gets screwed up. So there lies the importance of the translation of the right kind of gene at the right place at the right moment. Defects which are born out of these flawed genes in the nuclear encoded mitochondrial genes, are associated with hundreds of clinical diseases, phenotypes including anemia, dementia, hypertension, lymphoma, retinopathy, that's a disease of the eye, seizures, and neurodevelopmental disorders. Ophthalmic defects secure a very significant position in mitochondria associated health complications, and I'll explain it why in the following slides. As compared to most of the diseases related to heart, liver, brain, or kidneys, ophthalmic research is a sort of backbencher. The situation is improving and lots of vision research has come up, but still people has a tendency to think, oh, it's not a heart related problem, or it's not a kidney related problem, so I can sit back and relax. But the CDC mentions very strictly that at least 2.2 billion people have a near or distant vision impairment, and 75% of vision loss is still preventable and treatable with advances made from prior vision research studies. The better treatment of eye diseases reduces economic burdens to society and communities and contributes to a better safety. It's trying to educate people that even if it's a simple cataract problem or a vision problem, please do not sit back and relax. You never know where it can lead to. Mutations in the mitochondrial DNA are undoubtedly associated with a diverse spectrum of human disorders. More controversially, it has been claimed that they accumulate during normal aging and that they are responsible for specific age-related decline in bioenergetic function and tissue viability. It has been widely accepted that the overexpression of ROS can be induced in the surface of the eye as a result of many acute and chronic diseases and even in normal aging. So we see for hypoglycemia, which is an increased level of glucose in the blood, normal aging and the genetics of the person, even a rise in intraocular pressure can give rise to oxidative stress inside the mitochondria, leading to mitochondrial dysfunction, which leads to the formation of diabetic retinopathy, age-related macular degeneration, also glaucoma. Usually, the ROS-induced damage in the lens cell consists of oxidation of proteins, DNA damage, lipid peroxidation, and interestingly, all these three that I have just mentioned generates cataract. That's a very common eye disease. So in a nutshell, we can say that with abnormal cytosolic ROS accumulation, there is a mitochondrial damage because there is a mitochondrial depolarization. Now, what is the mitochondrial depolarization? So if you remember when I was explaining the functions of the different components of the mitochondria, we said that in the inner membrane resides the four complexes of the cellular respiratory unit. And these complexes, they maintain a proton gradient across the membrane. So that's what is called as a polarized mitochondria. So when these proton gradient is broken, hampered, the mitochondria becomes depolarized. Oxidative phosphorylation does not happen correctly. And hence, there is not only damage to DNA, but also the proteins and the lipids are undergo oxidative damage, as a result of which the cells progress towards apoptosis through the disruption in their DNA strands, which leads to pathological outcomes like there could be a rise in intraocular pressure, or we could lose retinal pigmented cells or the photoreceptors. I'll explain these two terms in my next couple of slides. And this progression of the visual complications can even have extended effects of Parkinson's, even include extended effects of the Parkinson's, which is actually a brain disease. Now, before I explain the strategies and a few data of my research over here, I would like to educate you a little bit on the different structures of the retina. Now, retina, as we all know, is the light sensitive part of the eye, and it results from an evagination of the central nervous system during embryo development. A small part of the retina is the most important site, and it's referred to as the macula. It's towards the back of the eye. It's only about 5 millimeter across, but it's responsible not only for the central vision, but most of our color vision and the fine details of what we see. There is another part in the retina which is called the fovea, and this is where the eye focuses the light. So when we rotate our eyes to see an object, what we are actually doing is we are actually rotating or moving the fovea to receive the focused light coming from the object that we are trying to see. Now, the retina is composed of different neuronal cell layers and the photoreceptor layer. The different neuronal layers have different functions. So one set, they convert light into electrochemical gradients. The other set of neurons, they receive and process the information from the first set, and they transfer it to the photoreceptors, to and from the photoreceptors. The third set, they send the processed information through the optic nerve to the encephalon, which is a part of the brain, and finally we perceive the image. The photoreceptor layer, this one, consists of cones and rods. You see the blue colored structure are the cones, and in between we have this little fat structure. These are the rods. The rods respond to light intensity, while the cones, they are responsible for perceiving colors. After the photoreceptor layer comes another layer called the pigmented epithelial, made up of epithelial cells, which are literally brown, carries brown pigments in it. And this is the outermost layer of the retina and are devoid of neurons. Now, these pigments are in these epithelial cells, they avoid light dispersion, thus helping in having more sharp vision. Since they are in close contact with the photoreceptor layer, they help supply nutrients to the photoreceptors, maintains this photoreceptor layer, helps in its biogenesis as well as homeostasis. Now, one of the most common of the visual complications that we come across is age-related macular degeneration, and I am certain that my audience has also heard of these common eye disease. It has two forms, the wet and the dry AMD. Now, a normal retina, like we saw in the last picture, consists of densely formed photoreceptors, the RPEs, the choriocapillaries over here, and a layer between the RPE and the choriocapillaries is called the Bruch's membrane. Now, underlining the choriocapillaries are the large choroidal blood vessels. What happens in dry AMD is there are waste products that collect beneath the macula, and they are referred to as drusen. The RPE cells are progressively lost. You see, there are gaps in between, and with disease progression, they get this loss is more severe, and some of the RPE cells may be damaged, and the choriocapillaries, these black dots here, are almost lost and degenerated. Moving over to wet AMD, you see the choriocapillaries are not there anymore. These blue colored cells, you see it's all degenerated. And because of the loss of the choriocapillaries, as we know that blood, oxygen gets supplied through blood. So this area is devoid of oxygen, and that's what we call a hypoxic environment, and this produce hypoxia inducible growth factors. The expression of hypoxia induced growth factors pulls in the expression of VEGF, which are vascular endothelial growth factor molecules. Now, the presence of these two growth factor types of molecules induces the formation of choroidal neovascularization, which means the formation of new vessels between the brush membrane and the RP. So they just push themselves, they force themselves between these two layers. As a result, the attachment between the RPE and the brush membrane that we see in a normal retina is lost, and the vision also gets impaired. Finally, with the disease progression, what we see is swelling, bleeding, scarring, and massive vision loss. From the clinical point of view, this is what we are looking at: a healthy eye and age-related macular degeneration diseased eye. So this is the macular part of the healthy eye, which is responsible for our sharp and color vision. And you see small accumulation of yellow colored lipid droplets. They are termed as drusen, and this is the healthy macula. So when we do some experiments with both the healthy and the AMD, we have to take help of a special machine called optical coherent tomography machine. And this is what the machine does. It can identify the individual layers of the retina. So in a healthy eye, we can see all the different layers of the retina by the machine. When we compare it with an AMD eye, I don't think so I need to explain what is the difference. It's all distorted and a disintegrated form. The last panel, here we see how we perceive an image through a healthy eye and what happens in the early AMD situation. And of course, you see how it deteriorates in the later stages. At this point, I think that we all have gathered some sort of idea about the cause and effect of an unhealthy, diseased system. My research here is aimed in connecting mitochondrial dynamics to RPE degeneration. In the next few slides, I will try to give you all a simplified view of how we are trying to connect the missing dots in order to achieve our goal. Interestingly, there are several reports where vision loss has been connected to neural problems. Few recent reports over here, they have shown how ophthalmologic problems are associated with neurodegenerative conditions, mainly AMDs connected to Alzheimer's and Parkinson's. So in this picture, we have a healthy eye that can go bad and present diseases like glaucoma, age-related macular degeneration, diabetic retinopathy, or retinitis pigmentosa. And on the right, we have the picture of a normal, healthy brain, which can go bad and show signs of Alzheimer's, Parkinson's, multiple sclerosis, and amyotrophic lateral sclerosis. We do think that there is definitely a connection between the diseases of the eye with the diseases of the brain, and the connector being the optic nerve. I work with a protein called DJ1, and the corresponding gene is referred to as PARC7. From the name itself, we can identify that PARC7 has something to do with Parkinson's, and yes, you are right. It was identified as an important gene in the Parkinson's disease. Now, what we are trying to do is to associate this particular protein, the DJ1, to RPE degeneration and also retinal degeneration under oxidative stress, and also to find out what's the connection of RPE degeneration with progression in Parkinson's disease. We also have a DJ wild type and a knockout or a mutant mice where the DJ is not present. We also work with these two mice models to validate our cell-based in vitro data. Due to Cleveland Clinic policies, I cannot show you all the experimental data, but I'll definitely try to share some of the research strategies that we follow to arrive at our conclusions. To start with the different types of cell line used for our vision research studies, but we are concentrated on the retinal pigmented epithelial cell line, or otherwise called as ARPE19. It's a spontaneously arising retinal pigment epithelia. That's how the name comes. And the number 19 is because the cell line was derived from the normal eyes of a 19-year-old male who died from a head trauma in a motor vehicle accident. The characteristic features of this cell is that they form stable monolayers even on porous filter supports, which we normally use for our experiments, since they exhibit morphological and functional polarity very neatly and nicely. The RPE plays a critical role in the development and maintenance of the adjacent photoreceptors in the vertebrate retina. In order to understand whether the cells are growing nicely and properly, we use RPE-specific markers, and we label the cells with these two markers and look for a nice fluorescing picture. So this is a very preliminary way to explain how we do our cell culture and our experiments. So this is a typical cell culture flask that we use, and using a very specific media containing fetal bovine serum, which is an important component to grow and develop our cells. So the cells grow on the base of this flask. And once the base is covered completely by the growing cells, we trypsinize the flask. That means that using trypsin, we dislodge the cells from the base of the flask, and then we distribute these cells according to our experimental requirement. The type of culture vessel that we use for our experiment is usually a rectangular plate that is made up of different wells. It could be 6 wells, it could be 12, 24, and so on. And each of these wells has a sort of a hanging plastic basket. So a well in a well, that's how the setup goes. And this hanging basket contains a very thin film of membrane, on the top of which we grow our cells. We feed the cells with the media that were used here and in the upper compartment and also in the lower compartment. When the cells are ready for our experiment, we treat the cells with 200 millimolar of hydrogen peroxide. Why do we use hydrogen peroxide? So our intention is to mimic the situation inside a cell when ROS is generated. And hydrogen peroxide is known to be one of the very commonly used ROS generators through the formation of hydroxyl radical. Finally, when the ARPE cells are treated with hydrogen peroxide, ROS is generated inside the cell, and what we try to figure out is what's the functional and structural damage that the hydrogen peroxide brings in to the mitochondria through specific experimental strategy. In the right side, we have a cartoon, which is I would say it's just the way we do our experiments. These are some AMD hallmarks which are detected in 1-year-old PGC1 alpha gene. Now, PGC1 alpha is connected to mitochondrial biogenesis, but as I mentioned earlier, that we do not use PGC1 alpha, we are concerned with DJ, the Parkinson's gene. If you look at this top panel, we see that this part, the left side, is of a healthy RPE with properly developed photoreceptor layer, the RPE layer, and also the Bruch membrane. These are the 3 layers that are specifically affected with AMD, like we have seen before. Now, with progression or onset of AMD, we find a atrophic RPE or a diseased RPE layer. We find degenerative changes on the photoreceptors and drusen deposition, which finally it leads to RPE death. These are some of the parameters that are studied with the disease. And the most important one is you see the number of healthy mitochondria, they go down rapidly, increasing the number of damaged mitochondria. This is the most important outcome of this disease, which points out clearly that mitochondria are greatly affected in a disease like this. And if we go back, think in the cell point of view, we have a huge accumulation of toxic products inside the cell that brings out this outcome. Going to our experiment, before I tell you what exactly we are looking at, let me explain this. Now, when oxidative stress inside the mitochondria it gets generated, mitochondria is affected first functionally and then structurally. Healthy or stressed mitochondria can be determined by fluorescent labeling. In the electron transport chain, as if you remember, there are reactions which gets generated continuously, leading to proton electrochemical gradient potential. Now, the active transfer of the positively charged protons generated in the inner mitochondrial membrane, it results in a net internal negative charge, and this is known as the mitochondrial membrane potential. So negative inside, positive outside. So of course, there is a difference in the potential, and that is termed as the transmembrane potential. Now, this net negative charge across the healthy mitochondria, it can be detected with positively charged dyes called tetramethylrhodamine, abbreviated as TMRE. This TMRE emits a red fluorescence. This is the mitochondrial labeling or the mitochondrial fluorescence that you are seeing. This is the black region is the nucleus, and you see this red fluorescing mitochondria across the nucleus. So the intensity of fluorescence, it depends on the transmembrane potential. If it's a high membrane potential, we get high intensity. If it's a low membrane potential, we get a low intensity. Having said that, when we look at our experiments, we have a control set. So you have seen how we set up our experiment. So we treat one group of cells with 200 micromolar hydrogen peroxide, whereas the other set stays as the control. We then label them with this TMRE dye and wait for an hour to see the results under the microscope. The graphical representation clearly shows that cells which were treated with 200 micromolar hydrogen peroxide shows a lower intensity than the control set, which clearly points out that with rise in oxidative stress or with the implementation of a stressed environment inside the mitochondria, the cellular respiration and oxidative phosphorylation is definitely affected, and it gets reflected through data like this. And of course, the mitochondria gets stressed. Another interesting phenomenon which is connected to the functional disruption is the structure of the mitochondria. Any change in the function affects the shape of the mitochondria. The mitochondria can be tubular like this, or it can be round or donut shaped, or it can be elliptical, depending upon the intensity of the stress that it has to encounter inside. They can form networks to maintain a connection with another cell and live happily, or it can undergo fragmentation, forming two cells from one cell, and in order to reduce the load of disrupted or disorganized mitochondria. To check the changes in our experimental strategy, we use a special labeling theme. Here we take the help of baculovirus, but don't get scared. This is a non-infective virus type frequently used as a gene transfer tool. Now, this engineered virus, it contains RFP, which is a red fluorescence protein, or we can also use GFP, which is tagged to an actin sequence because we want the fluorescence. This is a general idea that I am giving. I'll come to my specific experiment right after this. So the actin is the actin filaments are the area where we need this tag to be fluorescing. So what we do is we package the baculovirus with this fusion construct, and through an endocytotic entry, the fusion construct is released inside the cell. It travels to the nucleus. It is transcribed to the corresponding mRNA, gets released in the cytosol, and the fusion protein gets translated in the cytosol. And because of this actin sequence, the protein is directed towards the actin filaments where it fluoresces, where this red fluorescence protein comes into function. In our experiment is exactly the same design, only in place of actin, we use E1 alpha pyruvate dehydrogenase, the leader sequence for mitochondrial targeting. So we know that when this construct expresses inside the cell, it would 100% get targeted to the mitochondria where it would show its fluorescence. Most mammalian cell types are transfected with high efficiency, and even we use a viral mode of delivery, it's not toxic to the cell. And this transient transfection can be detected after overnight incubation for up to 5 days. So what do we see in our data? This is the control set of cells, and these are the 200 micromolar stimulated ones. For the control cells, we see the nucleus and nicely labeled mitochondria arranged at the side. They could be tubular, and usually they are in a tubular shape. In the stressed situation, first of all, what happens is the mitochondria travels to one side of the nucleus. So this might be more clear. You see the nuclear boundary here, and the mitochondria all traveling to one side of the nucleus. And it forms a sort of mesh over here. You cannot identify very easily whether it's a tubular structure, whether it's a round structure or elliptical. But you do see that inside the mesh, we do have an increase in the number of round structures, which shows that definitely the mitochondria are trying to fragment out to release the stress load. When we see such structural disorientation in the mitochondria, we know that there are two things that are happening: fission, fusion, shuttling, and mitophagy, which is autophagy of the mitochondria or cell death of the mitochondria. In the control set, like we have seen, the healthy mitochondria continuously fuse, forming tubular structures, and this helps in mixing lipids, proteins, and mitochondrial DNA. Now, the new components are added to expand and replenish mitochondrial networks through the mitochondrial biogenesis. Under a stress, one more thing is when we go for mitochondrial fusion, we should not forget of these three important proteins: mitofusin 1, mitofusin 2, and OPA1, which are actually responsible for maintaining the functional mitochondrial potential so that oxidative phosphorylation can occur nicely. It also helps to protect the mitochondrial DNA function and helps in cellular respiration. Now, under stress condition, the mitochondria usually gets fragmented. And that's when we start seeing the round or donut-shaped structures. The number of fragmented structures, of course, it depends on the power of the cell to handle the adverse situation, how intense the oxidative stress is inside the cell. When the mitochondria starts fragmenting, there are three proteins again that becomes very much functional. The mitochondrial fission factor 1, FIS1, and DRP1 that brings out a low or defective mitochondrial potential, almost non-functional mitochondria or damaged mitochondrial DNA, and a very low or damaged cellular respiration. Now, mitochondrial homeostasis is maintained by the expression and function of the fusion and the fission proteins. And this is how they preserve a healthy network of mitochondria. The unhealthy, unwanted mitochondria is not required by the cell, and what the cell does is it just kicks it out of the cell. And how does it do that? So an unhealthy mitochondria is first encapsulated by a double membrane autophagosome. The autophagosome then fuses with the lysosome, and it's called an autolysosome. This lysosome has acidic environment inside, and it contains degrading enzymes which degrades these bad mitochondria inside, and then whatever is left, the proteins, lipids, they are recycled back to the cell for mitochondrial biogenesis. But this is not where it ends. When we go deep into our analysis, we have seen that proteomic data from retinal pigment epithelium, or the protein analysis data from this particular set of cells, revealed significant changes to mitochondrial proteins. And importantly, those mitochondrial proteins were associated to glycolysis and TCA cycle, suggesting that potential functional defects and damage to mitochondrial DNA is connected very much to AMD progression. Fisher and Farrington have also supported this idea and the data that mitochondria in the RPE are a site of primary pathology for dry AMD. And this forms a rationale for our experimental study to target the mitochondria in the RPE as the most efficacious intervention strategy if administered early, so that we can prevent vision loss and cell death. In order to understand from the disease point of view, in order to correctly identify the source, we adopted another technology called the seahorse technology. So this technology is the first in vitro metabolic measurement which profiles metabolic activity of cells in minutes. It offers a physiologic cell-based assay system determining basal oxygen consumption, glycolysis rates, ATP production, respiratory capacity, and in a single experiment, it assesses mitochondrial dysfunction. I should mention here that this system is not only restricted to the animal system. Since plants also encounter stress, this system can be applied to analyze the stress effects in plants, the stress intensity in plants. And they are trying to come up with such machines as well. So here, to make our life easier, this machine, it measures the extracellular acidification rate called ECAR, which is a result of glycolysis, and also it measures the oxygen consumption rate, which is OCR, an indicator of mitochondrial respiration. We now know that the cristae is the site of cellular respiration, matrix the site for Krebs cycle or TCA, and the cytosol is the site of glycolysis. When we concentrate on this graphical representation, let me describe this to you. The blue part is what is represented as the ATP generated from the mitochondria, and the red part is the ATP generated through glycolysis. This is a control set, a treated set, and this is where glucose has been substituted for galactose to as a negative control of glycolysis. Now, if we compare between these three, we clearly see a very significant difference in the ATP generation through different sections. In here, the control set, we find the mitochondria is responsible for more ATP generation than glucose. Similarly, in the treated part, there is a slight increase in the glycolytic ATP, but it's not a significant amount. Whereas if you use galactose in place of glucose, you are not getting glycolysis, you are not getting any ATP generated through glycolysis, and the major part of it is mitochondrial ATP. So this forms the basis how the experiment is done. We have cells and we have some preliminary data using this experimental strategy, and we have seen that with 200 micromolar of hydrogen peroxide, which mimics a chronic stress situation because we are stressing the cells for 17 hours. So it's like a long period of stress. And we have seen that there are some amino acid precursors that are greatly affected, amino acid precursors of the TCA cycle, which are greatly affected. So the experiment is still under progress. Very recently, I am also connected to a project which is directly related to mitochondrial DNA, and this is about a DNA polymerase gamma, the replication repair system of the cell. POLG, as it's usually termed, the deficiencies cause mitochondrial dysfunction, and it promotes accelerated aging and tissue degeneration. So POLG is upregulated to protect the damage to the mitochondrial DNA induced by the increased superoxide or other ROS generating agents. But chronic exposure to superoxide radicals, it overwhelms the mitochondria or the replication repair system of the retina, and the damaged mitochondrial DNA, it propagates the vicious cycle of strand breaks and lipid peroxidation, protein oxidation, and finally the mitochondria, it becomes dysfunctional and initiating the apoptotic machinery, and we see RPE cell death. This is a very interesting slide, and we have got this information last week. We are amid the human coronavirus disease. We are all aware of that, and it's caused by the SARS-CoV-2 virus. But we were interested to find out, does SARS-2 virus have any connection with retinal degeneration, meaning RPE degeneration with COVID infection? A very recent publication from the lab group have shown that yes, COVID infection is definitely connected to retinal degeneration. So what we did is we took sections of the retina and we did two types of experiments. Firstly, we wanted to see how the retina is getting affected comparing to the control healthy sections. So if you look at the lower panel first, this is a toluidine blue stained retinal sections, and this is the control slide where we see all the sections or all the layers of the retina nicely arranged. Here we have the RPE and the choroidal capillaries, or the choriocapillaries. When we move over to the COVID-affected patient retina, we cannot even say which layer is what. They are all disintegrated, disorganized, and we see these choriocapillaries infiltrating into the RPE layer and breaking the connection. You see gaps in the receptor layer over here, and so you lose the connection and everything becomes a fuzzy slide. So now we thought, like, are these because of the system getting compromised, and or there are actual viral particles in the retina infecting the retina? We were not sure of that. So what we did is we took retinal sections and we labeled the section, the COVID, both the control and the COVID infected, with mouse anti-SARS-CoV-S protein that detects the virus if present inside the retina. And we do see the presence of the virus. These green spots you see in this COVID slide, they definitely speak of the presence of the virus. So actually, the retina gets infected with the virus. So it's not only lungs that gets affected with the COVID infection. And we were really excited with this outcome. American Macular Degeneration Foundation, they have, sorry, they have conducted studies that finds higher rates, excuse me, they have found higher rates of severe complications of macular degeneration in patients, which shows COVID complications or the other way around. So it's interestingly and definitely important to go into more further research of how to connect retinal problems, RPE problems, and moreover visual problems with COVID infection. So far, we have seen how mitochondria behave under this sort of negatively stressed environment. We also have gathered some idea about the cause and effect of oxidative stress, very preliminary ideas. But I think that it would be an injustice to this wonderful audience today if I do not mention one last very interesting feature of the mitochondria that has come up very recently. It is termed as mitochondrial hijacking, and it is applied basically to brain cells or glioblastomas, which are fast-growing type of central nervous system tumor. A new study has found that up to 20% of glioblastomas in its aggressive form are fueled by overactive mitochondria. These glioblastomas usually affects the brain more than the spinal cord, and it's a sort of terrorism in the brain. A particular cancer lab at Cleveland Clinic, they work very intensively on glioblastomas, and one of the lab members, while observing the progression of the disease, found out that there are cells. It could be neural cells, specific neural cells, or it could be microglial cells, which are also neural cells, or it could be the mitochondria itself leaving the diseased cell and walking into a nice one, not the dying cell, but a functional one to survive. So they are still working on it, but they call it as hijacking of the mitochondria. So in the diseased cell, the cell would ultimately die. So the mitochondria, in order to continue functioning, is taken up by an actual cell which needs more energy. And of course, we know that a tumor cell, in order to sustain, in order to have full control inside the body, has to function very effectively. So they are not yet convinced that it's the mitochondria walking into a new cell or whether they are microglial cells. So they are still researching on that. As promised, I would like to end my presentation here. And I believe that some of you would really vote for the fact, if not all of you, that mitochondria indeed inspires a lifestyle. I sincerely thank everybody for their patient hearing and would love to answer any questions. Thank you all and a very good night. Thank you.
Thank you very much, Dr. Bhattacharya, for your nice lecture. It's really enlightening session for us, especially for the students. Now it's time for the question answer session, as usual. And for this, I would like to request Professor Sanjeev Kumar Saha, Senior Faculty Member of the Department of Botany, to conduct the question answer session. And over to SKS, sir, please.
Am I audible?
Yes, very much.
Thank you, madam. It's really an illuminating lecture to us as well as our students. Now we shall move on the question answer sessions.
Sure.
We received many questions in our chat box. Among them, the first question is from Dr. Subhajit Bandyopadhyay, the faculty member of our department. The question is there any contributions of mitochondrial DNA in tracing evolutionary history of plants and animals?
Research has been going on. Definitely there is, because as far as research says, I won't be very able to say about the plant system, but for animals, yes. In the genetics department of Cleveland Clinic, they are working on this evolutionary aspect of mitochondrial DNA in combating diseases. So they are working on it, and there are evidences that they have reported, which shows that evolution have changed the DNA with disease progression.
Thank you, madam. Our next question is from Debashish Chakraborty. His question is there any role of metformin, drug of diabetes, to damage mitochondria in long-term therapy?
Metformin actually, over here, doctors are going against the use of metformin, because in some cases, they are not quite sure, but they are still researching on this. They have seen that metformin has side effect, and sometimes it's affecting the kidneys. So they are still researching on that, but the drug is still, it's there in the market, but they are trying to, or they are suggesting patients not to use it on a long-term basis. They are predicting some side effects on the kidneys.
Thank you, madam. Another question from our student, Pratyusha Singha. Does mitochondrial DNA determine phenotype?
Mitochondrial DNA, yes, because if your DNA sequence, so what happens, the DNA sequence is translated into protein. And the protein, of course, the expression of the protein would be affecting the phenotype. If you have any change in your mitochondrial DNA, it would get reflected in the protein expression, and the phenotype can change. Yes, it has an implication.
Thank you, madam. Another question from another students of our students, Pritam Banerjee. Can I say mitochondria as an independent cell of the early time?
It could have survived, but it chose a different path. It got fused with another prokaryote for its survival. But they, like I mentioned in my introductory slide, it can survive since it's the sole energy producing organism. It could have survived, but there is always this process of symbiosis which the organism are prone to adapt. So I would just state it this way. Yeah, it could have survived, but the situation didn't allow it to survive as a individual organism. The cell required its presence.
Another question from Rachna Chandra, and it's related, you see, that would like to know more about mitochondria fission, how it happens. Further, I would like to know about the role of mitochondria in retinopathy of prematurity?
Sorry, I won't be able to say much about prematurity. It's out of my research area. But definitely for fission, what I would say is the two proteins that I mentioned there, FIS1 and DRP1, also the mitochondrial fission factor 1, they are very effective in carrying out this. So the DRP1, so when a mitochondria undergoes fission, it first develops a constriction with the help of MFF1 and DRP1 on the outer membrane of the mitochondria. And this DRP1, it helps or it forces the mitochondria to break into or fragment into two separate units. And then comes fission 1, which is inside the mitochondria. So the fission happens this way. And once this mitochondria gets fragmented, if the cell cannot control the stress by itself, the autophagosome machinery gets activated, and then it engulfs the bad mitochondria and throws it out by the lysosomal machinery. That's how it occurs.
Thank you, madam. And last one is Rajesh Kumar. From Rajesh Kumar, what is the ratio of exons and introns in mitochondrial DNA?
Sorry, I have no idea about this. I cannot, I cannot answer.
Yes,
that.
madam. From my side, just a single question is there, that whether any, actually, how do the stressful life affect on the mitochondria, causing aging and Alzheimer diseases like these, or even the hormone affect on the mitochondrial function?
Okay.
You see that in we studied that, yes, most actually, I don't know that there's 99% genes, nuclear genes, which actually is responsible for mitochondrial component, and 1% is mitochondrial genes. And whether this mitochondrial diseases, this is caused only by these mitochondrial genes, like this.
Actually, no. There are some nuclear encoded genes also that brings in disease complication. So here, what we have seen, what I have talked about, are mostly the ones that are conducted by the mitochondrial genes. So, like you mentioned about hormones and how stress affects the mitochondria. Yeah, since all the cells in our body, they require energy for their survival, and our energy producing unit is the mitochondria. That's where the importance lies. Any of this cellular respiration, when we build up stress, when we get stressed, our the respective hormones that comes into play, the proteins that gets targeted with the change in the chemical sensors in the brain, everything is like sort of a crosstalk between them. So it's not like one particular thing, but it actually adds up to the cellular, to the mitochondrial, the biochemical processes that goes on inside the mitochondria. Even if it's like Alzheimer's, the main center is the brain. Even if it's Parkinson's, main center is the brain, but the neural cell also has its own mitochondria. So ultimately, it comes over to the mitochondria there, and it is affected. And once one of the hormones is affected there, or one of the proteins is affected at a particular site, it's not only that site that the protein is responsible or functioning for. Since there is a big crosstalk inside our body and everybody is communicating with everybody, one site that gets affected is sort of translated to some other site. So if you are stressed, you might get, like, let's say, a kidney problem, and we might worry, why is it? This is the reason. Like, where is the connection? So that's what my research is, but though I'm oriented for the eye part, but definitely
Yeah.
biochemical process inside the mitochondria gets affected.
Oh, actually, our lifestyle, whether our lifestyle may rectify this damaged mitochondria or it's protect the mitochondria from its damaging.
Definitely, our lifestyle can, if we try to lead a healthy lifestyle, because some of it is against us. Like we cannot control some things, like pollution, we can control, but everybody has to try together. But what is in our hands, like leading a healthy lifestyle, eating healthy foods, not smoking, things like this can improve the mitochondrial quality, can improve our cellular quality inside our body. Living a happy life, smiling, like, things like this, not to allow negative thoughts to overcome us. Things like this. Psychologically, we have to be very motivated and we have to stay very positive. What When talking to doctors here, I have seen that more than providing or prescribing medicines, they try to motivate us. They try to tell us that bring out your own solution, stay diverted from negative thoughts. So I very
Yeah.
much support that idea.
Yes. Thank you, madam. It's in the molecular, from the molecular level, we can say that yes, our healthy lifestyle can improve even the mitochondrial damage, or even it can protect from damaging of mitochondria. It's the hope for us, actually. So now I think this is very actually excellent seminar to us. Now I hand over the microphone to our HOD madam.
Thank you
HOD
so
madam,
much.
please.
Thank you, sir, for the nicely conducted, beautifully conducted question answer session. We have reached almost at the end of the session today. Now I would like to request Dr. Kuntal Chattopadhyay, respected coordinator, sir, of IQAC, Narsingh Datta College, to deliver vote of thanks. Over to
Thank you, Pompa madam. Can you hear me?
Yes,
yes,
I
sir.
can.
Yes, sir.
Okay, thank you very much. Indeed, it has been a wonderfully deliberated session. We have learned so many things in a very lucid and yet in a very sophisticated manner. We are really very delighted to have our respected speaker with us, and I acknowledge my heartfelt thanks to her.
Thank you so much, sir.
Thank you. Welcome. And I would also acknowledge thanks on behalf of IQAC to the Department of Botany for having chosen such a very interesting area of research to be talked about this evening. I also congratulate all the members of the IQAC for cooperating with this program. I must extend thanks to our team members in the technical domain who have always given us all kinds of support. And all the participants should also be thankfully acknowledged this evening, who are here this evening. And of course, before closing this kind of formality, so to say, I must acknowledge thankfulness to our principal madam for always standing by the side of this kind of an effort this evening too, and hope we will have such wonderful evenings in near future. Thank you all. And I am now handing over to our principal madam to close down the session with her observations.
Thank you very much, Honorable Speaker, for your fantastic presentation and lucid deliberation. I'm sure that all the participants, especially our students, they have got immense knowledge from your deliberation, and through their questions, they are now able to know what they were asking for. And I would like to congratulate Department of Botany for handling this kind of session.
Thank you.
I'm sure that in future we will be able to have some opportunity to listen to this kind of lectures again. And now I would like to again thank you all and good night. And now I declare the session is closed. Thank you.
Thank you so much.